segmented body of an annelid worm. So other really interesting annelids are those that live really deep in the ocean, thousands of meters deep in the abyss of the ocean, near these hydrothermal vents. And these worms are referred to as vestimentiferans, which this word kind of has the word vestiment in it, which means cloak or robe, and that kind of refers to these big tubes that they exist in. And these are just really fascinating worms. They can be up to 2 meters long and 4 centimeters wide. So we're talking about worms that are just huge, much bigger than any of the other annelids that we've already discussed. And they were first discovered in 1977 in ocean bottom rifts near the Galapagos Islands. But since then, they've been found throughout ocean bottom ridges. And again, they live near these hydrothermal vents, which means vents that are giving off these, that because of the openings in the crust of the Earth, they're emitting this hot seawater that is very rich in reduced inorganic compounds, such as hydrogen sulfides and methane. All right, so we have H2S and CH4. So compounds that are just really in high abundance in this hot seawater coming out of these vents. Okay, so these worms are really interesting, and they used to think they were in a completely different phylum, and then eventually they were put into the family Ciboglinidae, which is within the polychaetes of the phylum Annelida. So they're also just polychaetes, which is kind of interesting because they're so morphologically different. And of course, to live down there in the deep abyss of the ocean, you have to have some kinds of unique adaptations, which they certainly do. And one of the first things we can talk about is their morphology here. Their bodies are divided into these 4 main regions. They have this cephalic lobe where the tentacles are emanating, and this is an area for gas exchange surface. So it's just primarily gas exchange. We don't see a whole lot of feeding done with these tentacles, as we're going to talk about in a minute here. Then they have this little anterior section, which is has a lot of glands in it for secreting different kinds of enzymes and other stuff that we're going to talk about in a bit. And then they have sometimes this anterior section is referred to as the forepart. You might see that in some books. And then they have this really long trunk area, which includes most of the rest of the worm. And it has these papilia, cilia, girdles of setae. So we see a little remnants of something that looks like a polychaete to us. And then when you finally get back down here to the 4th section called the opisthothoma, which of course just refers to an abdominal part in arthropods, but here we're just talking about this last part of the worm, and this is the part here that reminds us of other kinds of polychaetes. It's the segmented area. They have septi to separate the metameres. There are setae. It has circular and longitudinal muscle. So unlike the rest of the worm, the opisthothoma is like, oh yeah, this is really a polychaete annelid, isn't it? So here's one of the more abundant of the members of this family, and it's in this genus Riftia, okay, the beard worm. And here you can see a close-up of the tentacle end. And recall that the worm that we just looked at is that they live inside these really big tubes, which again are several inches wide and 6 to 8 feet long. So these are enormous worms. And remember also that they live directly in the path of the vent outflow from the thermal vents. So their tentacles and probably the rest of their bodies are just being bathed in this hot water, okay, which actually becomes cooled by mixing with cold seawater. Okay, so the cold seawater down at the bottom of the ocean can vary. It's can be right near freezing, 0 degrees centigrade, 1 or 2 above, 1 or 2 below. The pressure keeps it from freezing down at that temperature, along with salt concentrations. But when it mixes with the hot thermal vents, the temperatures are about 10 to 15 degrees centigrade in the water, which is pretty balmy. That's about 50 to 60 degrees Fahrenheit. So that's pretty balmy for the bottom of the ocean, that's for sure. Okay, and so they live directly in the path of the vent outflow, and remember that the seawater is very rich in hydrogen sulfide and methane. Now, the trunk of this animal that we just talked about not only contains the gonads, but it contains a really unique structure which is multi-lobed, and it's called the trophosome. Okay, and the trophosome is the really important part of this animal in terms of how it gets its nutrition, because the trophosome contains densely packed bacteria. Okay, so you can have bacteria up to about 10 billion individual bacterial cells per gram of trophosome tissue. So they just have huge amounts of these specialized bacteria, which is how they're going to get their nutrition. Because the other interesting feature of these guys, unlike most annelids, all the other annelids that we talked about, is that they have no digestive tract. Okay, they don't have an esophagus and a crop and a gizzard and an intestine and a stomach and all that stuff. No mouth, no anus, no nothing. They have no digestive tract, which is an interesting difference from the rest of the annelids. And so how do they get their nutrition? So there's a few different ways that they can get nutrition. The tentacles themselves can actually do a little bit of external digestion of food particles if there's any around, but they really don't get much in the way of nutrition this way. Okay. Also, they can do a really interesting thing where they can take up dissolved organic matter directly from seawater. And because dissolved organic matter is going to be in low concentrations, they can use active transport and take up DOM against these low concentration gradients across the tentacles. And for some other species in the family, those worms that are called pugnophorans, this method here can meet a large portion of their needs. But for the vestiminiferans and some of the other members of this family, the primary mode of nutrition is going to be through a mutualism with these bacteria that we talked about in the trophosome, which are chemosynthetic. All right, so chemosynthesis is a really interesting process. So it's a lot like photosynthesis. And what's the point of photosynthesis? Well, chemosynthesis, like photosynthesis, can take small inorganic compounds and eventually build them into large organic compounds, which are the food molecules that they can feed on. So what's probably apparent to you at this point is that when you're down 5,000–6,000 feet at the abyss of the ocean, that there's not going to be any sunlight, right? Whoops, sorry about that. There's going to be an absence of sunlight. So photosynthesis isn't really an option. And the idea of utilizing chemosynthesis then is how these organisms can live, and not only live, but really thrive in huge numbers, in a huge size. So the bacteria in the trophosome will obtain their energy by oxidizing the hydrogen sulfide, right? So we're going to either oxidize H2S or CH4 in the surrounding seawater. So I want to just talk a little bit about what we're talking about when we say chemosynthesis here. And so I've rewritten this equation below because I realized that the subscripts didn't really come out when the PowerPoint got transferred to this program, but you can see it a little bit better here. And then the orange is to show that the equation is balanced. Okay, and this S here is sulfur, if you couldn't really read that. So what does this look like? Well, this looks like photosynthesis, right? You take inorganic compounds and you convert them into large organic molecules. CH2ON is the generalized formula for carbohydrate, right? And then eventually other macromolecules can be produced. So in order to accomplish this process of chemosynthesis, the bacteria in the trophosome are getting energy, again, through the oxidation of H2S and CH4, just in the way photosynthesis uses water, right? And so instead of H2O, we have H2S involved here. And so the bacteria can use the energy that they get from this oxidation, okay, in order to produce ATP eventually. So they oxidize these substrates in order to produce ATP, and then ATP is used for the synthesis of organic molecules. Okay, so instead of the energy from sunlight, we have the oxidation of H2S. All right, so again, very similar to photosynthesis. So it's a pretty amazing process. And what's really interesting is that we see that similar chemosynthetic bacteria have also been found in several mollusk species at these deep thermal vents as well. And it may actually be more common than we realize in animals that live in really deep water that are either really low or lacking in sunlight, or just in sediments that have high amounts of hydrogen sulfide or methane, even regardless of their temperature or depth. Okay, and so the bacteria then can use these carbohydrates for their own growth and reproduction. Okay, and then the bacterial populations, when they really build up, are then digested by the host worm. Right, so they can feed the host worm either indirectly by releasing soluble carbohydrates, or the bacterial populations just get digested by the host. A couple of pictures of some other individuals in this family. We saw Riftia, this is Tevnia. Here you can see the tubes kind of reflect that sort of segmented look, but this, again, the worm is living inside of these tubes. And here is a picture of Riftia outside of the tube. Again, that large sort of parchment tube that they live in. And this shows a close-up of that anterior portion where you can see some of the papillae and even some of the ciliation down here and the papillae up here. Okay, so that concludes our discussion of annelids. And if you have any questions, you can ask them in class. Thanks.